So, hi everyone. Thanks for coming. I'm going to talk about some collaborative work that I've been doing recently with some people in genomics at Princeton. And so we're interested in studying biological data, and I'll show kind of how we can use mathematical algorithms to actually analyze biological data and overcome some difficulties that they've had with the experiment. So I think it's a nice demonstration of how mathematics and computation can actually help overcome some obstacles that you have from the experimentation side of things. And I always preface when I talk about this work with, I am not a biologist, so this will be at a very low-level biology, and I don't know much about the experimental details, but I will tell you what I know. So the overall problem is that we want to reconstruct dynamics from snapshots. So if you're interested in studying developmental dynamics, there's two basic schemes you could think of. One, you could look at the development of one organism over time. So I take a fish out of a pond and I watch it grow up over time, and I can see how it develops. This can be troublesome for many reasons. Maybe the fish takes a long time to grow up, or maybe you want to look at things that you have to first kill the fish to look at. You can't look at it while it's growing. And so the second experimental scheme is called cross-sectional data. In this case, you take a snapshot of the pond. You have many fish. Each fish is a slightly different age. And so from these snapshots, you can actually reconstruct the developmental dynamics. And so then you're charged with the task of ordering the fish by their age. And also, if you have pictures of fish, you'd like to register them in some consistent frame of reference. So down here, you can see this is a schematic. Again, we have all our fish, and then you've registered them by where their heads are located and ordered them by size. And so now you can see clearly how the development progresses. So we won't be looking at fish, we'll be looking at Drosophila, which is a common organism people use to study developmental dynamics. Drosophila is the common fruit fly. It's about 1 millimeter in length. It's shaped like a grain of rice. And we're going to look at Drosophila during the 3rd hour of its development. So down here you can see some representative pictures of 5 minutes, 25 minutes, and 45 minutes into its development. And they've been stained for a specific regulatory protein. And you can see that the concentration profile of this protein changes dramatically over time. And the important thing is that we cannot look at this protein without first killing the embryos. So you cannot look at this in a longitudinal setting. You have to look at the data from snapshots. And instead of looking at the side view of this grain of rice, we're going to actually look at a slice down the long axis of the embryo. So turn the grain of rice on its head and then look down, and you see that now this red stripe that you see along the side now expressed at one peak, and there's a axis of symmetry in this embryo, so you actually see another peak on the other side. And so thanks to some experimentation ingenuity, you can collect lots of data of this type relatively easily. So using a microfluidic device that people at Georgia Tech developed, you can actually collect many, many images with relative ease. So here you see, this is a picture of Drosophila, and these are the pictures I've been showing you before. And here you see this microfluidic device. So essentially what it is a long labyrinth of channels, and you flow a solution of fluid with the embryo suspended in it through these channels. And along the sides of these channels, there's these little traps, and each trap is sized so that one embryo can fit per trap. And so the fluid flows in, and there's a pressure drop, and so it forces one embryo per trap. And once there's an embryo in the trap, it closes this little channel behind it, and so no more embryos can go in. So what you get is you get an array with one embryo pulled in per trap. You can see this here. They stand up on their heads, and then you just put this whole device into a microscope, and you can actually image each embryo one by one. And so what you end up getting is images, cross-sectional images like I showed you before, about 90 microns in from the poles of the embryo. And although you can control all the embryos so they all go on their stand upright, you can't control the rotational orientation of the embryos within the trap. So what you end up needing to do is actually rotate all the images to be in a consistent frame of reference. So this is a typical data set that people would collect. You can see each image is from a different embryo, and it's been stained for 2 different proteins, the red and the green. And each embryo is fixed at a different developmental time and in a different rotational orientation. And we don't know the correct time or the correct rotational orientation for each image. And so I'm going to show you how we can actually automatically both register, this means rotating everything to be in a consistent frame of reference, and temporally order these images. So to go from this array of images on the left to the array of images on the right. And you can see on the right, all the green peaks are now pointing down, and you go from essentially no red signal to a very pronounced red signal towards the end. And this can all be done automatically without any prior knowledge of the biology of the system. So when we first started this project, basically the person who does the experiment sent me these images, and I was told I was supposed to order them. So I knew nothing about what I was even looking at. So the first task of image registration, we're going to use a technique called angular synchronization. And so the problem is that we want to register the images to be in a consistent frame of reference, but we don't know what good image features are. So if I told you I wanted to register a bunch of faces, you'd probably say, okay, well, I can find the nose and the eyes in everybody's face and then sort of match everybody together. But I don't know what the, quote unquote, nose and eyes are for these images. And you also don't know what a good template is. So maybe if you had a representative image, you could just shift everybody to look approximately like the representative image. But if you pick a really bad representative, then you're going to get very bad information. But what you can do is compute pairwise alignments. So down here, instead of having images, now I just have little arrow vectors. And you can compute the angle you need to rotate one vector to another, or rotate one image to another. This doesn't require any template, any features. You just see how to rotate pairs. And from there, what you want to do is you want to take these pairwise measurements and extract the best rotation for each image. So you want to take this array of pairs and then find how you want to rotate each image so that they're somewhat consistent. And you can actually show that under some conditions. You can stack the pairwise measurements into a big matrix. And then if your measurements are approximately correct or only corrupted by some nicely behaved noise, then the top eigenvector of this matrix can actually give you the best rotation for each image. So it basically boils down to an eigenvector problem to find the optimal rotations for your images. And so I told you the second task we want to solve is the temporal ordering. We want to take our snapshots and order them in some way so that we can understand the developmental dynamics. And we're going to use an algorithm called diffusion maps, which is a general manifold learning technique. So what we're going to assume is that our data points, in this case our images, lie on some one-dimensional curve. So essentially, although the images are very, very high dimensional, right, that each one has many, many pixels, the development is such that it goes along some one-dimensional, perhaps nonlinear trajectory in this space. And so if we can extract a parameterization of this data or parameterization of this curve, we can order the data along it and then essentially have our images ordered in time. So again, this is a schematic. You can see data points in 2 dimensions, but each data point lies on this one-dimensional nonlinear curve, and we like to automatically extract this parameterization. And again, this is going to boil down to a different eigenvector problem. So what you do is you take your data and you calculate the pairwise distances between pairs of data points. And you assume that points that are close in the original space should be close in your parameterization, right? If I have 2 images and they look very, very similar, I think they should be close developmentally. And so then you put all these sorts of ideas in a big matrix. So you use some kernel of the distance, which means the value will be large if the data points are close, and it'll go to 0 if the data points are not close, if the images look very, very different. And then you find a parameterization where things which are close originally are now close in your parameterization. So I told you about 2 algorithms with both boil down to eigenvector problems, and you can actually combine them into one step called vector diffusion maps. So this algorithm will both find the best rotation, the best alignments for all your images, as well as order them in time. And it works better than doing each step sequentially because you don't try to align things that are dynamically very different, and so it's more robust. So these are the results, these are the original images, and then these are the ones that have been registered and ordered using this algorithm vector diffusion maps. And like I told you before, the green peaks are all nicely aligned, they're all pointing down, and the red peaks grow in intensity as a function of time. So I lied to you slightly in that for this particular system, both the registration and the temporal ordering can be done using prior knowledge about the system. So the registration, like I've been pointing out, the green peak specifies the ventralmost point of the embryo, and so you could align everything just by where the green peak is. And the embryos can actually be timed by another protein. So you can stain for these membranes that grow inward as a function of time. So you can see they're up here in gray, and this red bar indicates how thick they are. So as a function of time, they get thicker and thicker, and you can actually draw a calibration curve. So this is thickness of these membranes as a function of time, and so you can take each image, and if you also have the corresponding membrane stained, you can say how old the image is. So you can note that although this curve looks fairly nice, there's noise, especially towards the end of the development when these membranes stop growing. So we can compare the concentration profiles around this. We want to compare the two results, the results I showed you using vector diffusion maps and the results that we obtain just by registering using the green signal and then ordering using these membranes. And the simplest way to compare is to look at the concentration profiles around the periphery of the embryo. So for each image, we can unwrap the concentration around the circumference into a concentration profile. And then we can stack them all in an array. So down here, each row in this array corresponds to an image, and it's, the signals are going around the circumference of the embryo, and they're ordered by time. And so so the left is using the prior knowledge that I showed you on the previous slide, and the right here is done using vector diffusion maps, so automatically with no biological knowledge. And you can see the results are fairly consistent. You can also compute the rank correlation, which is basically how good the two orderings are, or how similar the two orderings are, and you get rank correlation of about 0.92, which is fairly high. So this was done, like I said, basically as a proof of concept for a data set that we already knew how to order using biological knowledge. But following this time period that I showed you previously, which is called cellularization, there's gastrulation. And this is what happens about in 20 minutes. So you can see that now we don't have this nice uniform arrangement around as a circle, but things start to morph and deform. And during this time period, there's really no good way to time the pictures. There's nothing like the membranes that grow inward to let us say how old each image is. But you can see there are some nice patterns developing, and these things turn in and eventually form the future organs of the organism. So here you see a larger data set taken during this time period, and we'd like to actually do the same thing, register and order. So we did this, and you can see this is kind of hard to process, but if you look carefully, it looks fairly nice. And what we actually did to make it simpler to digest is compute an average trajectory. So for each chunk of about 10 images or so, we computed the average image, and that's what you see down here. And if you compare it to the previous slide, it looks fairly similar. The gray signal looks fairly similar, but you can see now the red and the green are also on here. And again, the red and the green are things that you cannot look at in live images. You have to kill the embryos before you can look at them. So this gives us a very nice picture of what's happening during this time period, and it was not known previously or is not easy to measure previously. So in conclusion, we have looked at cross-sectional data and how we can automate the tasks of registering and ordering cross-sectional data from biological experiments. And we've used mathematical techniques to do this, and that don't require much prior knowledge about the biology of the system. And we were able to validate the algorithms we were using in the first example, where we had independent time and space markers. And in the second example, we got relatively consistent results with what we expect. And so right now, what I'm actually working on is merging these pictures with live movies. So although in these, you cannot measure the green and the red in live movies, you can actually measure the gray. That's how I got the pictures here. And so we're interested in sort of merging the two so that we can not only order the data, but actually get a good time for each of the images. And then we can look at not only the developmental trajectory, but also the kinetics of processes. So I'd like to thank CSGF and Krell for funding research. And with that, I'd like to take any questions.